For any help regarding biology nursing assignment help, visit https://www.nursingassignmenthelp.com/. Email info@nursingassignmenthelp.com. Call us +16786484277. Do your reading, review your homework and lectures. For questions B and C, use reference material and don't be afraid to think outside the box. You may use the images in the PowerPoint file 20.20113 question to structures.pptx to draw structures for your answers. Use PPT drawing tools to decorate the images. Then select the image and its associated decorations, copy, and paste in your Word document answer sheet as a PDF or JPEG image. Alternatively, submit the PPT file with your answer document. Hypo 1 was found to be toxic, so the chemists came up with Hypo 2. You have been hypomericillin 2 OCH3F, hypomericillin 1 OCH3F, HO NNSOOOOONOHN. Asked to predict whether 2 will be more or less toxic than 1, or about the same. Ignore the changes in bond angels, it's my mistake. Back up your answer with chemistry, including what you think is the most important electrophile for each structure. B. In preclinical toxicity testing of diclofenac in Chinese hamster ovary cells containing the entire complement of human cytochrome P450 metabolic activity. F. Nursingassignmenthelp.com. The drug was only mildly toxic. When all of the P450 enzymes were killed, inactivated, by exposing the cells briefly to carbon monoxide, CO, the drug was even more toxic. What was going on? C. In testing for DILI in vivo, it is common to monitor blood for an aminotransferase, example, ALT, AST, etc. But this fails when the drug causes cholestasis, and testing for alkaline phosphatase is the preferred assay. Please explain. Nursingassignmenthelp.com. Question 2. When thinking about toxicity, we can think about drug metabolizing enzymes, that are xenobiotic encounters during both phase I and phase II of metabolism, which generally consists of hydrolysis, reduction, oxidation, and conjugation reactions. If the drug encounters an enzyme that can convert xenobiotics to reactive electrophilic metabolites, these metabolites can therefore react with nucleophiles in the cell such as DNA or proteins and covalently bind, leading to toxicity. The enzyme most often associated with the process of generating reactive electrophiles is cytochrome P450. My approach therefore for this problem will be to analyze how many sites in each drug can be recognized by a metabolic enzyme and subsequently converted to an electrophile. A. Structures and approach are attached on a separate PDF file. I outline the possible reactive site in each molecule and which I think will form the most reactive intermediates. Hypo 1 can form the following potent metabolites: nitrogen cation electrophile, a sulfinic acid, an orthoquinone, and an epoxide group, and a ketone intermediate. Hypo 2 can form the following potent metabolites: a paraquinone, an alpha, beta-unsaturated gamma-decarbonyl compound, an epoxide, and a ketone intermediate. It seems as though Hypo 2 could be less toxic than Hypo 1. Because you are losing the toxicity from the heterocyclic tertiary amine oxidation and the sulfur oxidation, but you still have major quinone toxicity. I would say that the most important electrophile for each structure is phenol rings, which can easily be converted to quinones. Quinones are powerful electrophilic Michael acceptors that can react with cellular nucleophiles and form protein adducts. For example, Neurotoxicity associated with Parkinson's disease can be explained by paraquinone formed from the oxidation of dopamine, Labinsky et al. 2009. Nursingassignmenthelp.com 1. B. Diclofenac is an NSAID or nonsteroidal anti-inflammatory drug which is used to reduce inflammation. This is its chemical structure. Diclofenac can get extensively metabolized into well-characterized metabolites including 4-hydroxydiclofenac, 5-hydroxydiclofenac, 5-OHDIC, 3-hydroxydiclofenac, 4,5-dihydroxydiclofenac, and 3-hydroxy-4-methoxydiclofenac. 4 and 5-hydroxydiclofenac are known to form highly reactive quinine imines. Without cytochrome P450, usually diclofenac is less toxic in vitro because there is no formation of the reactive metabolites. In this case, however, diclofenac has some toxicity associated with itself. Diclofenac in vitro has been shown to impair ATP synthesis by the mitochondria, Bort et al. 1999. Impairing ATP synthesis would essentially cause cell death. While diclofenac's metabolites can also impair ATP synthesis to some extent, usually the metabolites are present with some concentration of the parent molecule as well. Here, we just have a large concentration of diclofenac that can all react with the mitochondria and essentially is not titrated away by ACYP. On the other hand, diclofenac could be metabolized by another enzyme in the cell. UDPGTs, for example, have been shown to react with diclofenac to produce a reactive electrophile that can form protein adducts. From Professor Tannenbaum's Drug Metabolism to lecture. Perhaps we do not normally observe as much of this reaction because diclofenac binds with a higher affinity to CYPs than UGTs, but in the absence of CYP we can observe this effect, assuming CHO cells express UGTs. Nursingassignmenthelp.com. C. Cholestasis is a condition where bile cannot flow from the liver to the duodenum. In testing for drug-induced liver injury, according to the FDA's standard of H-Wise law, a drug that shows hepatocellular injury will show greater than 3-fold elevation of ALT or AST above the normal and greater than 2x the upper limit of normal for total bilirubin levels. However, since cholestasis is an obstructive condition, it can also cause increases in serum bilirubin levels, but it has not been characterized to cause high elevation of ALT or AST. Therefore, by just testing for ALT and AST, cholestasis will be overlooked. However, testing for ALP levels is more specific to cholestasis over general DILI. Since in cholestasis, the bile ducts can become blocked, ALP levels will rise in the serum because it is expressed in the edges of the cells that join together to form the bile ducts. In general, hepatocellular injury-induced necrosis can vastly increase the serum levels of ALT and AST. However, cholestasis is functionally a blockage disorder caused by inhibition of A transporters such as BSEP. ALT and AST will not be majorly transported into the serum during cholestasis. Sources: Bort, R. et al. Diclofenac toxicity to hepatocytes, a role for drug metabolism in cell toxicity. Pharmacology. 1999-88, 1. Riggin, S.L. Ethyl acyl glucuronides, the good, the bad, and the ugly. Biopharmaceutics and Drug Disposition. 2010 31, 7. http://laptist/sunline.org/understanding/analytes/tab/test. Nursingassignmenthelp.com. Hypomericillin to hypomericillin 1. S. Ho. NOO.